going again. Here we go. All right, so that PCR video was me setting up the PCRs. Then they move on to the thermocycler. My reaction is, I think it's on the order of like an hour and a half. There are definitely faster PCR reactions that you can do, but it's where that thermocycler is going to send the DNA to replicate through many different cycles and replicate those specific regions that the primers basically dictate. And when I send it off for sequencing, this is called a fragment analysis, and we'll see what that fragment analysis looks like. So I do this analysis in a program called Genius, and the reason why I use Genius is simply because it visualizes the file types that I get from the sequencing center. Oh, it's advancing on its own now. Let me just fix that. There we go. So when I get my samples in Genius, the first thing that I have to do is align the ladders. So as we talked about, there are 3 different dyes that get incorporated into each reaction, and each reaction is going to be sequenced separately by the sequencing core, or the genomics core. They are going to add a dye into my samples that acts as the ladder. So this ladder has different peaks at these different size ranges, so that all of my samples can be aligned to one another. In this case, you see that over here, you might not be able to see very well, that it was a GeneScan 500, that there's no 250 BP peak, and that the dye Liz was used, or LIZ, and that it's an orange dye. And so when I first got my samples back, the Genius program is going to try and predict where the peaks are. It doesn't do the best job, and so I have to go through each of my samples and say, the program would say that these 2 are peaks, and so I have to go through and delete them, add peaks where there are peaks. But you can see that at the end, you end up getting all of your samples nicely aligned to each other. And in this program, Genius, there are many different settings. This program does everything. It is not a free program, and there's a reason for that. I scale my x-axis so that all of my samples are aligned to one another. I have made my x scale and y scale smaller so that it all fits into the same screen, but there are all sorts of manipulations that you can do with each of your samples using this program. So, once my ladder is there, then I can go through each of my primers. You can see that over here, this is where the different dyes can be selected. You can have 4 dyes sent to the center and visualized in Genius. You can see that it's 6-FAM, and I talked about hex and rock, so why is it VIC and PET? That's just because the program read them differently, but you'll see it doesn't matter. And then I go to each of my primers and I call my genotypes. So it's hard to see, but this up here says SO mother 104. This says LO male 162, SO male 163, offspring 1, offspring 2, offspring 3, offspring 4, and offspring 5. This is what the outputs on the module are going to look like. And what you're going to have to do is you're going to have to call the genotypes. So most of them have the numbers beneath them. Sometimes when genotypes were close together, the program gets unhappy and says, no, these are the same peak, and you just have to guesstimate what the peak actually is. But we can see that the mother here is heterozygous up here at 200.1 and 202. So as we follow down, one check that we should do is that every offspring should inherit 1 allele from its mom and 1 allele from its dad. So all of these offspring should have around a 200 peak. We see that this one has a 199.6, rounds to 200, 200, 200, there's a 200 there, 200, 200. So good, our offspring do seem to match our mother in that sense. Then what we're going to do is look at our different males. This male has a 198 peak, this one has a 215 peak, and then this one is homozygous for 200. Now something that you'll have to think about in your module is when a male has the same genotype, or the same allele as the mother, how can we tell if the offspring belongs to that father or if it belongs to, or if you're just seeing the sort of remnant of the mother. So that's something to think about, and one of the main reasons why we have 6 primers instead of 1 primer is for this reason exactly. Often individuals in the same population are going to have the same alleles at this locus. So anyway, you can go through, call your alleles. As of now, it looks like this offspring belongs to this father because they both have the 198 peak. none of the offspring inherited the 215 peak. These are just some interesting things when you get your output for your particular cross to look at. So basically I went through each die, set the expected range, so I told it that this primer in particular, or this locus in particular, had this amplicon size range and ensured that all of the peaks landed in there. Did the same thing for the green die and the same thing for the red die. Now what you're going to get is in this case this is reaction A, you're going to get trace results like this, which show you your red, your blue, and your green all together. You're going to call the individual alleles, you're going to write them down in the module. So there's also a reaction B, but this gives you an idea. These are 3 of the 6 loci that we're looking at. In Genius, this is something that I'm not going to give you, but in Genius it actually does tell you the genotypes themselves, but it's good practice for us to go through here and look at what the numbers actually look like. It's also kind of a callback to the old days in which you had to look at a gel output and determine this information. So what I've done is I've viewed and recorded all of the alleles for each locus, and then I use a program called Service to verify paternity. Service actually takes all of this information and uses algorithms to determine the maximum likelihood that a father is the actual father of a particular offspring. In this case, we're doing sort of just a rough look at this. And so just like in other labs where we've had different gels that you could get, it's going to be different mothers and all of their offspring. There are 6 possible ones that you could get. And so as for the module, what you're going to be asked to do, set up the PCR, right, like the other ones, look at the trace outputs, determine who you think is the most likely father for each embryo, and then answer a couple questions based on that information. And then it's really important for me to acknowledge sort of everybody that contributed to this project. So even though I presented as my work, I have a big lab that gives me a lot of brain ideas for these sorts of projects. I have an advisor, Dr. David Resnick, who is a faculty member here. He might be a good one to write your letter to. Our lab always has openings too if you think you're interested in working with fish. I have a lot of funding sources who help fund this project, and then I also have a lot of undergraduate volunteers who help me care for fish, help me do my DNA extractions. All of these things were done by undergraduate volunteers, and a couple of which from this particular class, which has been really awesome to see everything come around. So with that, that is pretty much all I have, but I'm happy to answer questions about any of this.
Okay, thank you, Samantha. Any questions, anybody?
And obviously I can't see the chat, but I would imagine that there may be chat.
Nothing in the chat so far.
Did you know that there were fish that have the functional equivalent to a placenta?
No, I didn't.
Right? They are really neat. While you're thinking about questions to ask, I'll just hold them up again. And I don't know if we could change the focus, but there is actually a baby in here. Let's see if I can flip them around. There we go. There's a baby right there. And so you might be able to guess if they're placental that they give live birth. And there are lots of really cool aspects of their reproduction that I didn't even go into.
Cool.
I have a question real quick.
Okay.
I'm pretty sure you answered this before while you were presenting it. So are the 3 primers for just determining the different genes, or the different in the smaller offsprings and the large offsprings?
Yeah, so these primers were actually not developed for this particular species. And so we don't actually have a particular gene that they're linked to in this species, which is kind of odd. It's not necessarily the best way to go around it. About it, we just know that there are many different, it's essentially that there are many different genotypes for these particular locus or loci.
Okay, thank you.
And we'll go over that a little more when we go back to class.
Yeah.
It's just kind of a weird question, but you said they have placenta, so like, are they like humans where they have to like deliver the placenta afterwards, or does it like when they give birth to their fish, does it just like what happens to it?
Yeah, that's a really, really good question. And the way that they have a placenta is so totally different from the way that humans do. In that, so when humans, oh my gosh, I'm forgetting my human reproduction. I'm going to explain it the fish way first, and then maybe Dr. Burnett can fill me in on the other side. So instead of the egg bursting out of the follicle actually stays around the egg as it's gestating with in a fish. So the fish is actually, the egg is inside of the ovary, and that ovary acts as the placenta. So this is usually easier if I draw it, but I don't know how easy it is to draw on here. It's not. So there's an ovary with an embryo inside of it that's covered in the follicle. In human reproduction, the egg bursts from the follicle and then travels. So in this case, there's no actual placenta, but we don't know exactly how it works either. The way that we think it might work is that the ovary is sort of just this little like nutrition soup, and that the mother is allocating resources into her ovary that then gets distributed to all of her offspring. But the follicle itself is also attached to the ovary. So there may be specific ways for a female to allocate to one individual offspring. And I know that probably sounds confusing because it really is, but it's interesting how it might work. But so the mother does deliver resources to her offspring, and she does deliver the embryo, and when the embryo is born, that's when it bursts from the follicle, essentially. And so that follicle acts like the placenta that would surround the baby.
So it remains part of the ovary, and the baby fish swims free.
Yeah.
Got it.
And they're born like eating and things, so they're...
Oh, so they're precocious.
Yes.
Wow. Cool. Good questions. Any other questions? Now's your one chance to ask about animals in this class.
Yeah, right. Yeah, we should have mentioned that. It's plants from here on out.
We're going back to rice next time. So, and so this is different from guppies, right? In that guppies don't have this placental thing, but they do give live birth.
Yeah, so what's really cool about guppies is that the moms are basically just an incubator. So what happens in guppies is they make an egg, and that egg has all of the resources the baby needs. So it's just like you could think of an animal that lays eggs. That baby has everything it needs. So what's really the point of the incubation? Well, there's actually a lot of advantages to giving live birth. You don't have to make any sort of calcified hard shell. It's swimming with you. As long as you're safe, your babies are safe. So the moms are basically just these swimming incubators, and then they give birth to the live babies, just like this fish does. But in this fish, the mom instead has the advantage where she can make really tiny eggs and then give it resources as she goes. And this can be advantageous for more than just the way of she doesn't have to make big eggs, but also if her environment is either really rich in resources or really poor in resources, she has that time to adjust. So either her baby doesn't get as many, or she can perhaps terminate the pregnancy for some of them, which does happen, and then they reabsorb the embryo. Or if they're in a really food-rich environment, they can just give their babies as much as they possibly can.
Cool. Yeah, and if you ever buy guppies, plan to have babies because they will usually come pregnant.
Yes.
We found that out when we bought some quite a while back, and then of course they mate in the tank.
Yeah, and that's also what makes this experiment difficult. If you're checking on your fish every day, nothing is really too difficult. But as soon as you see that there's a male in that tank that looks like it might be a male, you have to take it out. Because what happens is they actually inseminate females via a fin that becomes calcified, and then they use that fin to deliver sperm to the female. But that fin isn't calcified for about 4 days. And so once you see that fin is starting to elongate, you take those males out to ensure that the females are virgins for whatever experiment you're using or doing. I'm full of weird fish facts. Yeah, and also we can share my email. I have my Twitter up there at placentalfish, but my email can be shared too, or it's sleve004@ucr.edu.
So you keep fish at home
Yes.
and mate them?
Yeah, I mean, I don't do artificial insemination at home.
Yeah.
I don't. No, I mean, I've always been a super huge fan of fish. We breed goldfish and koi and betta fish. We have lots of fish at any given time.
Wow.
And I do mean it when I say that if you decide you want to fish and you want to email me asking about keeping fish, I'm happy to help. Because we have plenty.
Okay. All right. No other questions? Then we'll pause and let's say take a 10-15 minute break, and we'll start back in our usual Zoom links at 10:15. Okay, and we'll break down some of what Samantha went over this morning. Okay, so see you back 10:15 in your respective Zooms. All right. Thank you, Samantha.
Thank you. Bye, everybody.